This visualization compares the 180 liters of blood plasma the human body filters every day against the 1.5 liters that actually exit the body as urine. The kidney functions as a high-volume fluid reclamation plant. It processes the body's entire plasma volume, roughly 60 times a day, extracting the fluid to selectively reclaim the exact water and solutes the body needs to survive. The functional unit responsible for executing this massive fluid turnover is the nephron. There are over 1 million of these microscopic structures within each kidney. Each nephron is powered by two distinct biophysical engines. The first is a mechanical filtration system that uses raw hydrostatic pressure to force plasma out of the bloodstream. The second engine is a countercurrent multiplier, a paradoxical arrangement of tubules designed to selectively reclaim 99% of that water and solute. By examining the physics of these two systems, we can map exactly how the nephron engineers highly concentrated urine from raw plasma. This diagram of the renal corpuscle shows the site of the first passive engine. Filtration here is a physically driven process requiring no metabolic energy. Note the architectural contrast. Blood enters the capsule through a wide afferent arteriole and exits through a much narrower efferent arteriole. This arrangement of a wide input and a narrow output creates a high-resistance vascular bottleneck across the capillary tuft, known as the glomerulus. This chart of filtration forces shows that the structural bottleneck artificially drives the glomerular hydrostatic pressure to 55 millimeters of mercury, forcefully pushing fluid out of the blood. That outward push is resisted by two inward forces. The first is the hydrostatic pressure of the fluid already filling the capsule space, which pushes back at 15 millimeters of mercury. The second resistance is colloid osmotic pressure. Large plasma proteins remain in the blood and exert a 30 millimeters of mercury pull that attempts to draw water back into the capillaries. Subtracting the 45 millimeters of inward resistance from the 55 millimeters of outward pressure reveals a net filtration pressure of exactly 10 millimeters of mercury. This 10 millimeter margin is the sole driver for the entire glomerular filtration rate, yielding approximately 125 milliliters of filtrate every minute. Because this pressure margin is so narrow, the kidney's intrinsic autoregulation systems must defend it. If these systems did not constantly adjust arterial diameters to counter fluctuations in systemic blood pressure, the filtration engine would fail. After 65% of the fluid is bulk reabsorbed in the proximal tubule, the remaining filtrate enters the medulla to be deeply concentrated. This cross-section shows the baseline environment of the kidney cortex. Before the concentration process begins, the interstitial fluid sits at an isotonic state of roughly 300 milliosmoles. To break this isotonic state and create a salt gradient deep in the medulla, the nephron utilizes a countercurrent multiplier within the nephron loop. The multiplier depends on two strict permeability rules. In this diagram, the descending limb is highly permeable to water but traps salt inside. The ascending limb follows the opposite rule. It actively pumps salt out into surrounding tissue, remaining completely impermeable to water. These opposing permeability rules set the physical stage for a self-reinforcing cycle that multiplies the salt concentration of the inner kidney. The cycle begins when the ascending limb utilizes ATP to actively pump sodium chloride out of the filtrate and into the medullary tissue. This accumulation of pumped salt raises the local osmolality of the surrounding medullary tissue. This salty environment creates an osmotic pull that draws water out of the adjacent descending limb. Because water is constantly leaving, the filtrate remaining inside the descending limb becomes increasingly concentrated as it travels toward the hairpin turn. The multiplier effect occurs when this ultra-salty, concentrated filtrate rounds the hairpin turn and is delivered directly back to the ascending limb. This pre-concentrated fluid provides the ascending pumps with even more salt to push into the interstitium, reinforcing the initial effect. This continuous physical feedback loop is the driver that pushes the concentration of the deep renal medulla to an extreme 1200 milliosmoles. Standard capillary blood flow would normally absorb and wash away this delicate 1200 milliosmol gradient. The kidney avoids this using the vasa recta, a specialized capillary network that acts as a countercurrent exchanger. Blood flows through this vessel in the opposite direction to the filtrate. Its walls are freely permeable to both water and salt. As the blood dives into the deep medulla, It equilibrates by passively absorbing salt and losing water. As the blood travels back up toward the cortex, the process reverses. It absorbs the reclaimed water and leaves the salt behind in the deep medulla. This structural alignment allows the blood to carry away reclaimed water while leaving the 1200 milliosmol medullary gradient intact. This 1200 milliosmol gradient represents stored energy. Its utility is dictated by the body's moment-to-moment hydration state. During overhydration, the walls of the collecting duct remain impermeable. This allows dilute 100 milliosmol urine to pass through the medulla unaltered. In a dehydrated state, the posterior pituitary gland releases antidiuretic hormone, or ADH, into the bloodstream. ADH triggers the insertion of specialized water channels called aquaporins into the membranes of the collecting duct. As the collecting duct plunges through the 1200 milliosmol medullary gradient, the high osmotic pressure extracts water from the filtrate through these open aquaporins. This extraction results in the final excretion of a very small volume of highly concentrated metabolic waste. The kidney uses physical architecture and fluid dynamics to engineer thermodynamic gradients, maintaining fluid homeostasis across a wide range of external conditions.